Welcome back everyone. So far we have completed DNA replication process, enzymes, then we have seen DNA replication semi-conservative, then today we will see transcription process. Okay, after transcription we will go into translation, then after translation the protein will be formed. Okay, see, for the formation of protein it has to undergo first transcription, then translation. Okay, so in brief, when we talk about the formation of protein, the first process is transcription. In transcription, what happens? The DNA will give information to mRNA. That means mRNA will carry information from DNA template. Okay, we have already discussed during mRNA, but still here we are going to talk about in details. So in transcription, what happened? The mRNA is going to carry information from the template strain of the DNA. Okay, information means genetic code. So from transcription, mRNA will form. That means mRNA will carry information. Then after mRNA, if it will undergo into the process of translation. Okay, then it will undergo translation process, and finally after translation the protein will form. So this overall process we call it as a central dogma. First, for the formation of protein, the mRNA will carry information from the template strain of DNA. The process is known as transcription. We will talk now. Then after carrying information by RNA, it will undergo into the translation process, where all the genetic codes which are carried by RNA, all the genetic codes will get bind with activated amino acid, and there is some complex processes we will discuss in translation. Okay, then after translation, finally protein will form. Now this overall processes, transcription to translation process, we call it as a central dogma. Alright, so today we will discuss transcription. Transcription is important from your board exam point of view. This year also question came, and for entrances, you know, the DNA replication, the enzymes, transcription, translation, and then the genetic codes, then tryptophan operon we will discuss, leg operon, all those are very important from entrance point of view. See, from your board exam, the question will come in between of DNA replication process. Okay, the question may ask from any part from the replication process. So study all, clear your concept. The question will come from the short point of view, short point like Okazaki fragment, lagging strand, leading strand, primer. All that question may come in between of the topic for 1 or 2 more. So be careful. So study everything. Today we'll discuss transcription. All right. So first of all, we will see some important term. Then we will go the process of transcription. See, in prokaryotic, cell, okay, first of all, for the transcription to occur, an enzyme again plays a very important role, just like in DNA replication. There is also an enzyme which plays a very important role for the process to occur. These enzymes are RNA polymerase enzyme. Now RNA polymerase enzyme has many types again, but in case of prokaryotic, the RNA polymerase enzymes are only one in number. That means a single RNA polymerase enzyme helps in the process of transcription, where in prokaryotic cell. Prokaryotic cell mane bacterial cell burot. Okay, in case of bacterial cell. So in prokaryotic cell, single, we're going to write some important features or some important terms, okay, of transcription. Then we will go into the process. Single RNA polymerase. I'm writing short form polymerase. So single RNA polymerase help in the process of transcription. I should not be writing all this sentence, but let me read. In the process of transcription. Okay. Just like DNA replication, where different enzymes help in the process of replication of DNA, just like that in transcription process there are RNA polymerase enzyme. Okay, but the RNA polymerase enzyme has again many types. But in case of prokaryotic, it has only one RNA polymerase enzyme which help in the process of transcription. Okay, in prokaryotic. In eukaryotic cell, that means in case of our cell or higher living organism cell, what is pro and eu? Pro means early forms. Okay, karyon, this is the word karyon. Okay, karyon means nucleus, so early nucleus. Eu means later or modern nucleus. So this was the first evolved cell, this was the modified form of cell. Okay, so eukaryotic, we have discussed I think in first year. So in case of eukaryotic cell, that is in case of our cell or higher living organism cell, three types of RNA polymerase enzymes plays a very important role in case of transcription. Okay, there are three types of RNA polymerase enzyme found. Three types of RNA polymerase enzyme helps in the process of transcription. Okay, so basically for the process to occur, that means for the transcription process to occur, RNA polymerase enzymes plays a very important role. That's thing you remember. Okay, for the transcription process to occur, RNA polymerase enzyme plays a very important role. While in case of prokaryotic cell, there is only one RNA polymerase enzyme which plays or which function for transcription process. While in case of eukaryotic, that is in case of our cell, it contains three RNA enzymes. Okay, it has three RNA enzyme which helps in a process of transcription process. So we're going to see that. Number one, you write complete sentence in your own language. Okay, जो समझ में आते हैं उसको लिखना अपने से खुद से, ठीक है, लेकिन पॉइंट होना चाहिए उसमें. So there are three RNA polymerase enzymes which help in the process of transcription in eukaryotic cell. The first RNA polymerase enzyme are known as RNA polymerase 1. Okay, RNA polymerase 1 होते हैं, RNA polymerase 2 होते हैं, and RNA polymerase 3 होते हैं. So RNA polymerase 1, what they do, they help in the synthesis of rRNA. It synthesizes rRNA. ये सबका रोल है. We will see the role of rRNA, the role of tRNA, the role of mRNA. All those RNA are required for the process of translation and transcription as well. So we're going to see that one by one. So for the synthesis of rRNA, it is done by RNA polymerase 1 enzyme. Next, we have RNA polymerase 2 enzyme. Okay, it doesn't only synthesis rRNA, it also synthesis some rRNA, but this will do at this stage in a high secondary level. If you read only this thing, it is enough. It also synthesis some rRNA, okay, like 5.AS rRNA, then 18S rRNA, 26S rRNA. So those RNA also they synthesize. But at this stage, if you write only rRNA, this will do enough. Okay, otherwise you will get confused. But still, if you want it, you can write it. Okay, it is there in your NCERT book. But the most importantly, polymerase 1, RNA polymerase 1 help in the synthesis of rRNA. It's really difficult to pronounce rRNA. It's really hard. So RNA polymerase 2 enzyme, it synthesize or help in the synthesis of mRNA and hnRNA. What is hnRNA? We will discuss. I will show it when we discuss the process of transcription. It is heteronuclear RNA. HN means heteronuclear. Okay, so I should have shown this in mRNA also, but I left that hnRNA. But today in this topic I will show it. During the formation of mRNA strand, okay, the strand which is formed, the mRNA strand which is formed from template strand of DNA, the first formed mRNA strand is known as hnRNA. I didn't mention that when I discuss mRNA, but today I will mention, so you add those. Okay, so later on, wait for now. So this RNA polymerase 2 help in the synthesis of mRNA and hnRNA. Say from entrance point of view, all those are very important. Which enzyme synthesis what? If you want to clear medical, these things are important. There is one more. RNA polymerase 3 enzyme. RNA polymerase 3 enzyme, it also synthesizes. It synthesizes. Why I am writing off? It's synthesis tRNA. Now tRNA means transfer RNA. Transfer RNA. All those RNA has a role, a specific role in translation process. We will discuss this. Okay. rRNA, it is a ribosomal RNA. Ribosomal RNA, it helps in the synthesis of protein. It helps in the assembly of two ribosomes. Okay. For the formation of protein, for the synthesis of protein, the assembly of ribosomes are required. Ribosome has two subunits, smaller subunit and larger subunit. In case of eukaryotic cell, the ribosome are 80S, the larger subunit are 60S, smaller are 40S. Okay, for the formation of protein, those two ribosomes has to join or has to fuse. For the fusion of ribosome, rRNA helps. It's really hard to pronounce this, rRNA. Okay, whatever. So this rRNA helps in assembly of those two ribosomes. Okay, then the mRNA is going to carry the information from template strand of DNA. The informations are genetic code. We have discussed genetic code. The codes are read in triplet form. We have 64 genetic code. Out of 64, 61 codes for amino acid. Remaining 3 codes are anti-codon or nonsense codon. Examples are important, you need to remember. Then tRNA, transfer RNA. It is going to carry all the activated amino acid along with it to the ribosome. So we are going to see that. Okay. Today we will discuss transcription. Transcription, that is from DNA strand, the mRNA, how the mRNA is formed, or how the mRNA carries information from template strand of DNA in details. So these are the 3 enzymes which plays not 3 enzymes actually, these are the RNA polymerase enzyme which helps a very important role during the process of transcription, and they consist of RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Each RNA polymerase has a specific function, so those are the function they perform. Remember, that is in case of eukaryotic cell, that means in case of our cell. While in case of prokaryotic cell, that means in case of bacterial cell, the RNA polymerase enzymes are single, only one, which help in overall process of transcription. And this single RNA polymerase also synthesis all those RNA, okay, rRNA, mRNA, tRNA in case of bacteria. All right, okay, let's move further. Sometimes this question asks, describe the RNA polymerase enzyme in case of eukaryotic cell. So, you can show it like that nicely. So basically, which enzyme helps during transcription process? The RNA polymerase enzyme helps, okay. So apart from this, the RNA polymerase enzyme also contains some subunit, many subunit I should say. Apart from this, the RNA polymerase enzyme also contains subunit, because it is again a large molecule. Molecule means enzyme, a large enzyme. It contains subunit. Each subunit has a specific role in transcription process. Okay, so I'm not going to write sentence everything in here. So according to your idea or whatever you understood the sentence, write it by your own according to your comfort, okay. But the point, those points should be there, but the sentence according to your comfort or whatever you understood or whatever you want to write it, you write it by yourself. It doesn't matter. This is not an English subject or history subject. It is a science subject. Whatever sentence you write, it doesn't matter. The points should be there. Okay, so apart from this, they also contain some subunit, many subunits. So we're going to talk about one by one. So let me rub this one, alright. So we are talking about eukaryotic cell only, okay. We are continuing eukaryotic cell in case of eukaryotic cell. Prokaryotic में सिर्फ एक ही होता है, हो गया बस खत्म उसका. So in case of eukaryotic, we are continuing in case of eukaryotic cell only. So subunits are, number A, the first subunit are 2-alpha subunit. 2-alpha subunit. See, you have to remember, okay, you have to remember 2-alpha subunit. It has a role again. Let me write first all the name of subunit. Then the another subunit is known as 1-beta dash subunit. Then next we have 1-beta. This is beta dash, not prime, okay. 1-beta subunit. Prime words are used only in case of DNA and RNA. A, B, C, D. There we have one more. Epsilon naught subunit. I hope you know all those symbol physics. You have already studied all those, right? So 2-alpha subunit, 1-beta dash subunit, 1-beta subunit, and epsilon naught subunit. All those subunit has specific role to play during transcription process. 2-alpha subunit, what it do? It binds to initiation protein. You will understand when we discuss the process, the actual process. Okay, for now write it down, this function. See, again, just like DNA replication, before going to the actual process, first remember the name of the enzymes along with their function. Then you will understand the process. Okay, so 1-beta dash subunit, it binds to template strain of DNA. I know you are already having some idea when I'm writing this sentence, because you all are very smart and talented student, right? So you already understood this. It binds to the initiation of protein, okay? Then it binds to the template strand of DNA, who 1 beta dash subunit, okay? When the mRNA is formed from the template strand of DNA, this subunit of RNA polymerase is going to bind where? In the template strand of DNA, okay? So for the process to occur, the enzymes plays a very important role. There are so many enzymes which plays a very important role. 1 beta subunit, what it do? It binds to nucleotide. I know you are getting my point. I know you are smart. Then we have epsilon subunit. It helps in proofreads of mRNA. When a new strand, that means when the mRNA carries information from the template strand of DNA, when the mRNA strand are synthesizing new nucleotide, if there is something, by chance, if there is any wrong nucleotide has been added in mRNA strand, that nucleotide is checked by epsilon naught subunit, just like DNA replication. If there is any wrong nucleotide, it is going to replace it with the correct ones. See, because you know, if a wrong nucleotide has been synthesized, it is going to result in mutation. All right. So all those subunit we call it as a core enzyme. Okay. So RNA polymerase enzyme contains core enzyme. But how many number? There are 5 in number. So they contain 5 core enzyme. Okay, remember this. The RNA polymerase enzyme, apart from RNA polymerase 1, 2 and 3, they also contain many subunits like 2 alpha subunit, 1 beta dash, 1 beta and epsilon naught subunit. All those subunits are also called it as a core enzyme, and there are 5 in number. 5. So 2 alpha subunit, 1 beta dash, 3, 1 beta, 4 and epsilon naught 1, 5. So 5 core enzyme. 5 means it doesn't mean like that 1, 2, 3, 4. It should be 4. So 5 means 2 are here. 2 alpha subunit. मतलब 2 alpha subunit होंगे. And then 1 beta dash and 1 beta and epsilon naught. Okay? So all those subunits are also called it as a core enzyme. Core, C-O-R-E. Okay? Core মানে ভিতৰৰ আৰু so tak আমি core enzyme বুলিও কওঁ. So কিমানটা থাকে? 5টা থাকে দেই. All right. These are core enzymes. So we are talking about subunit. Okay? Now apart from this core enzyme or this subunit, they also contain again some subunit. Very large enzyme, RNA polymerase, DNA polymerase, they are very large enzyme. It contains many subunits. You can't even imagine what are the things happen inside our cell. What are things happening inside our cell? It is not easy to make a new organism. It is not easy, okay? It is not an easy process to build a new life. It has to undergo many complex things. Can you imagine? Though it looks simple, inside, only our cells knows what they have to go through. Okay? What am I supposed to say now? Okay. Apart from this subunit, they also contain other subunit like. So I am continuing after D, then E, then we have sigma subunit. Sigma subunit, okay. But all these 4 subunits are called as core enzyme. The sigma subunit Do not call it as a core enzyme. Why I am writing male symbol? This is not the sigma, this is male. Please don't distract me. Okay, this is subunit. Sigma subunit. Sigma subunit, what it do? It binds to RNA polymerase enzyme. Okay, polymerase I am again writing in short form. It binds to RNA polymerase enzyme. Okay, sigma subunit. Again, you have to know all the functions and the name of the enzyme before going to the process of transcription. Alright, then only you go to the process of transcription. All those has a specific role. You will understand when we go to the transcription process. There's one more. They are all important. You have to remember from entrance point of view. They are very, very important. Very, very important. The question may ask from any of this. So you have to remember its specific function. Okay. So there's one more known as rho factor. These are all subunit. Okay. Subunit of what? Subunit of RNA polymerase enzyme. So rho factor. What it do? Rho factor. It elongates. Okay. It elongates and initiate. Let me write first. It initiate and elongate mRNA. Okay, it initiates and elongate mRNA. See, when mRNA strain is formed from DNA strain, the elongation of mRNA, suppose this is mRNA, this is DNA strain, the elongation of mRNA, okay, this is the elongation of mRNA. The mRNA will elongate, slide over. Slide over means they're going to synthesis from template strain, right? When they slide over or when they elongate, this elongation is done by rho factor subunit. Okay. Now, one important thing you need to remember: the sigma subunit and the rho factor are attached only when they are required. Once their work is done, they're going to false off. Alright? They do not attach all the time. They are going to bind only when they are required. Okay? So that's you remember. They are attached only when they are required, and once their work is done, they're going to false off. Alright, so these are the subunit of RNA polymerase enzyme. Now in exam, for 3 marks, the question asks sometime, describe the RNA polymerase enzymes in case of eukaryotic cell. Prokaryotic mein nahi puchenge. So then you will write, in case of eukaryotic cell, RNA polymerase enzyme are of 3 types: RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. The RNA polymerase 1 help in the synthesis of rRNA. RNA polymerase 2 help in the synthesis of mRNA and hnRNA. RNA polymerase 3 help in the synthesis of tRNA. Apart from those, they contain many subunits. What are those? 2 alpha subunit, 1 beta dash subunit, 1 beta subunit, and epsilon naught subunit. All those subunits has a specific role, and they are 5 in number, and those subunit we call it as a core enzyme. Okay, so again they contain sigma subunit and rho factor. Those two are the subunit attached only when they are required. Once their work is done, they falls off. It has a specific role again. Okay, so these are the enzyme. Remember, study in a systematic way. First, study the enzyme with its function, then go to the process of replication. Not replication, transcription we are talking about. So next we will see the process of transcription.